on the reef site. There was an average of 11 samples as well used in each species group to get an average of carbon percent by dry weight. After the samples had an established carbon percent by dry weight, I then withdrew the remotely operated vehicle and diver video survey footage and utilized that in the MSJ software. I also set the scale to a known metric. Here we use the railroad tie that has a known width of 4.1 centimeters. And I isolated this portion of the reef site in MSJ, and I was able to calculate the known surface area. From that, I measured all the species groups within this photo and got their percent cover within this photo. And calculated the percent cover total by surface area structure. For each site, we used 5 of those photos or still frames minimum to calculate the percent cover. And then that percent cover per species was combined with the known surface area to get the total species surface area at each site. So that covers the encrusting organism protocol. We did have one organism that presented a diverse set of challenges due to its unique branching pattern and it being 3D in nature, and that is seaweed corals. So instead, I grouped these into size classes based on height and established a population per size class. These were my 3 main size classes: small, medium, and large, and I established an average of carbon grams per individual to estimate the total carbon present of these species. After processing these physical samples to analyze them, I separated them into structural material categories as well as reef profile patch height for analyses. I did this by utilizing light cyclone 3DR software and or known measurements of the reefing materials and split them into low profile, medium profile, and high profile reef sites. The results of my study for species groups, I was able to establish 8 different species groups ranging from bryozoans, barnacles, stony coral, sponges, biofilm, seaweed coral, sea fan coral, and snowflake coral. The carbon content was in fact significantly different among all species groups, with a significance factor of 0.001. I want to point out that bryozoans and seaweed coral as well as sea fan coral had a significantly higher carbon content by dry weight compared to other samples. And it is also important to note that barnacles, biofilm, and sponges, while they may have a lower carbon content by dry weight, they did dominate in number, therefore still capturing a significant amount of carbon when extrapolated to reef-wide analysis. Profile height significantly affected the carbon content per meter squared, with the most significant being between high profile and medium profile reef sites, while low profile versus high profile and low profile versus medium profile were not significant. Material type did not significantly affect the carbon content of these samples, so while there may be some correlation, it was not significant enough. This is the average carbon grams per meter squared by site type. So essentially, this is the carbon concentration that has accumulated on each site in square meters. You may
and environmental factors. So key predator and prey species were selected either based on their prevalence from literature or through the correlation test if there's a significant relationship exists. So we use the state space model to track population dynamics. So the true population size of blue crab at any given time can not be directly observed. So we could say it's a hidden state variable, and sampling data was just the observation, and it doesn't reflect the real population. So by fitting the model to this observation, we can simulate a simplified ecosystem that includes both biotic and abiotic factors. So the model can capture both internal trophic interactions and also external environmental factors. So this model is a equation base for estimating population dynamics. Right, the first parameter is the true abundance at time step t. So what we do here is to estimate how species abundance interact with each other over time. And we have a big community matrix. So that's describe how species interact with each other over time. And we have the C, that stands for covariance matrix. That's going to tell us the effect of environmental covariance. And we have U that accounts for delayed ecological effects from the environmental covariance, and H right here is we use it as a time lag. And E right here represents the process error. So it's kind of like random variation in the prediction process. So this error could be unmeasured biological factors or environmental variability. And the format kind of like so this is what we format matrix. So since the communities matrix is symmetrical, and I'm just showing the lower triangle right here. The row is the species, are the species being affected, and the columns are species external and effects. The main diagonal shows how each species is influenced by its own past abundance, and the off diagonal represents how different species of the interaction between different species, or interaction between covariates and each species. So each letter could represent the strength or direction of these interaction. Yeah. And the second part of this model is the observation equation. It explains how the actual data are collected, reflected the true population. So right here, Y represent the true observed abundance. C is the identity matrix that links the true abundance to observations, and V is the observation error. We could think it as sampling error or missing data. So our preliminary result of the early stage blue crab in Galvin Bay shows hard head catfish exerts strong precipitation pressure on blue crab. And in contrast, we do see positive interaction between red drum and oyster with the blue crab abundance. And we also observe dissolved oxygen and positive impact on early stage blue crab. So that said, this model currently shows sign of overfitting and possible of autocorrelation. So we need to we plan to refine the model and to improve the prediction. So traditional models also often assume linear relationship and fixed equation, but right here we all use empirical dynamics modeling to identify non-linear relationship that nonlinear relationship drives the population decline. So first we kind of like construct a state space to apply this nonlinear method. So this red line here on the left is the a single time series data, and it's just the projection of this complex system shown on the left, shown on the right. So this system includes many variables, so that we cannot like measure directly. So this model allowed us to reconstruct the underlying dynamics of this system using a single time series. So we take single time series as a vector x, and so at each time step t, we create this vector to represent the states of system at that time, and then we can use d-dimensional to represent the numbers of lags that can represent the complexity of this system. Therefore, as time moves forward, each of these vectors will become a point in this three-dimensional, multi-dimensional space, and then all these paths will become to a geometrical shape. So we can see it as the attractor. That will be the dynamics of this complex system. And by once we construct the state space, we can use simplex projection to identify nearest neighborhood and forecast future crab abundance. And we can apply SMAP to identify, to detect nonlinear interaction. And we can also use that to capture how the stress and interaction between variables changes over time. And after that, we can use convergent cross mapping. So this method is used to test for causal relationship between variables. So we can, so if a predator species significantly affects blue crab population, then we should be able to reconstruct the predator's population dynamics from the blue crab data. So for the future work, we will continue developing and comparing both models for all the blue crab, all sides of the blue crab with different year. And in addition, we will use both modeling approaches to forecast future blue crab population trends. And we can, by doing that, we can inform management decisions. So I would like to send to all the financial support and data access from TPWB. And I would like to thank to my co-author, Anderson, Zach Olson, and my advisor, Dr. Thank you. Any questions?
Yeah, we have 4 minutes for question. Okay, thank you. Let's move to the next talk by Melman Mill. His talk will be on metabolic advantages, predict interspecific competitive outcomes, symptomatic blending units.
Gonna shuffle, gonna take just a sec.
Yeah.
So
Bring it home. Thanks for coming today, everybody. Can you all hear me? So today I'd like to talk to you a little bit about metabolic rate and competitive ability in 3 species of local Texas blennies. I want to start by thanking all of the people that contributed to this project. An awful lot of grad students, technicians, and other various friends around MSI helped with fish collection, and then also quite a few people and funding sources on advising it. This project could not have been done without all of your help. So I want to start by sharing a scene that plays out in my kitchen just about every morning. Black cat is Cricket, and the brown one is Nutmeg, and they are wrestling over literally nothing. They just do this. They're sisters, you know. And Cricket generally tends to win. She's not a lot bigger than Nutmeg, but I think personally a big part of the reason she wins is she's got more stamina. She can fight harder and for longer, right? And so there's an innate energetic trait to her biology that can help predict who gets the preferred bed, who gets the preferred spot on the cat tree. She usually does, right? And so you could say this innate physiological trait of these cats is, to an extent, structuring their spatial community within my kitchen in my house. Unfortunately, there's not a lot of literature on relating physiology to behavior in cats, but there sure is in fishes. So the metabolic theory of ecology holds that the rate at which an organism's metabolism proceeds underpins all processes within that organism's body, right? And that rate can vary between species, between individuals within a species, and also over time within an individual. This bar represents a hypothetical range of metabolic rates of which an individual fish is capable. Their maximum metabolic rate, or MMR, is up at the top, and their standard metabolic rate, or SMR, is down near the bottom. The reason it's near the bottom is because if their metabolic rate were to fall below that SMR, they need that energy for cellular maintenance, things that must go on or the organism would die. And the difference between the two is called the absolute aerobic scope, or AAS. And that represents the portion of their energy budget that is for optional expendable activities, right? So let's say our fish gets into a fight with another fish. Maybe now it reshuffles its energy budget so that a greater portion of its aerobic scope is going towards activity, biting, swimming away, that sort of thing. But what if its opponent, for whatever reason, happens to have a lower NMR, and as a result, a reduced aerobic scope? They have less energetic capacity to put towards competition, and probably they would lose. The literature has quite a strong basis for this sort of connection between metabolic rate and competitive ability and behavior in general. There's quite a few studies that show that individual fishes with higher SMR tend to be more bold and take more risks when foraging. And in the last couple of decades, research has started to show correlations between larger aerobic scope and competitive ability in certain lab trials. But these studies usually fail to take into account what's actually happening in situ. And so that's what I wanted to try and address with this study. So here's where we are right now. This is UTMSI, that is us. And way over here is our nice little research pier. And it's suspended on these 12 big concrete pilings that are covered in these blenny communities. These blennies are an ideal study system, not just because they're conveniently nearby, but they're ideal for this sort of study because they're so site associated. If you dive down and you pull a blenny away from that piling, they're going to swim right back to it. They're constantly encountering each other. So they're almost certainly competing for resources, space, what have you. And also because they are cryptobenthic, short-lived fishes, which means their generation time is very short. So if there's any sort of nuanced population structuring that's happening over generations and iterated by evolution, it has certainly had the time to persist at this point. So here are Hyplurichyllus multifilus and Hyplurichyllus ocpennis, and we wanted to ask 3 questions of these species. First, at the species average level, do these metabolic traits, MMR, SMR, and aerobic scope, and various behavioral traits relating to competitive ability vary amongst those species? Second, at the individual level, is there a particular metabolic phenotype that's associated with their ability to win competitions? And then lastly, do the dynamics predicted by these lab studies hold true when we actually go and see how they're behaving on the pilot? So to measure metabolic rate, I use a technique called intermittent flow respirometry, which is a complicated way of saying we measured the rate at which they consume oxygen as a proxy for metabolism. On the left here, you can see the glass chambers where the fishes sit, and we use a series of fiber optic oxygen probes to measure their consumption rates. And here's what we found. So all of these graphs are mean mass adjusted metabolic rates. So this is milligrams of oxygen consumed per gram of fish mass per hour. So this is SMR. Doesn't look wildly different. We do have a significantly different spacing between Aquapenis and cristata, although I would say it's probably largely driven by this upward skew of several individuals. Although for those individuals, having an elevated SMR might mean that they have higher basal energy intake requirements. But we start to see bigger differences when we move to maximum metabolic rate. We're starting to see a little bit of a stair-step pattern. Aquapenis is significantly lower than the other two species. Multiphilus and cristata are broadly comparable in terms of range, although the maximum for multiphilus is higher. And then when we combine those two metrics, because again, aerobic scope is the difference between the two, we start to see a more direct stair-step pattern. There's a significant difference between multiphilus and aquapenis, and multiphilus is reaching higher highs overall and going over a greater range than cristata. So qualitatively, we could say that, at least on average, perhaps multiphilus is going to have a bit of an energetic advantage in terms of a larger aerobic scope. But what does that mean when we throw them in a tank and let them fight? So we conducted a series of dyad experiments. Here we have a multiphilus on the bottom attacking, I believe this is a scartella, and doing a pretty good job of it. You can see that we can separate these attacks into a series of discrete rapid approaches or attacks, and we can also filter out avoids, which are the opposite, sudden bursts of swimming away from the opponent. So we can quantify attacks per minute and avoids per minute for each individual fish. And we can take the difference between those two and have that be that individual's net aggression. So an individual that's attacking a lot more than they're avoiding has a high net aggression. An individual that's avoiding a lot more than attacking, which is going to have a low negative net aggression. However, because an attack by one individual does not always necessarily mean an avoid by another, they might just both be attacking or one might ignore the other. Sometimes you'll get studies, you'll get dyads where both individuals have a positive or even a negative net aggression. So to really directly compare them, you need to take the difference between the two to yield this individual's aggression differential, and then flip those terms around to get this individual's aggression differential. So what we've yielded here is an inherently definitionally reciprocal number where the winner is going to have a positive number and the loser is going to have that same number but negative. And so now we can construct models that predict behavioral outcomes both in terms of a binary winner-loser outcome, but also in terms of aggression differential itself, which takes into account both who won and who lost, but also quantitatively by how much they won or lost. So when we're looking at those binary fight outcomes, multiphilus tends to win an awful lot. The way this plot works is we have arrows pointing from winners to losers. You can think of it as the flow of aggression by these fists. So you can see that multiphilus only lost on 4 occasions in the entire study. Scartella only won 3 times, poor little thing. So just qualitatively based on this, I would say multiphilus is tending to win the most, aquapenis an intermediate amount, and Scartella the least. But does that trend hold true when we look at aggression differential directly, the actual quantitative levels of aggression? And the answer is yes, definitely yes. So this is aggression differential across all 3 species, regardless of who they're competing against. Multiphilus has significantly higher aggression differentials than both other species. This 0 line again demarcates winners versus losers. So you can see there's only 4 multiphilus losers that are below that line. Not only are most of the multiphilus above the line, but several of them are above it by a lot. They're winning by a fair amount. They are chasing aggressively. And then you might ask, well, what happens when we take into account who they're competing against? So this is the same graph, but separated out to include opponent. And if you look at the 2 rightmost values, you'll see that there's really no difference at all depending on who multiphilus is fighting. They are comparably very aggressive. I want to caution against overinterpreting other columns in this graph because it is, necessarily reciprocal data. I've included it for completeness. But yeah, so this, to me, pretty much answers our first question. On average, multiphilus is tending to have a fairly high maximum metabolic rate, greater aerobic scope, and they're tending to win these dyad competitions, and they're winning by a lot. But is that the actual mechanism? Is this elevated aerobic scope the actual mechanism that predicts their ability to win competitions? So we constructed a series of models that include species identity as a random effect to try and look at specific individual fishes. And the answer is pretty much yes. If you look at this left graph, the x-axis is the absolute aerobic scope differential. So it's the focal fish's scope minus their opponent's scope, right? How much of an advantage they would have in theory. And the y-axis is the binary win-loss outcome. So the focal fish that win are up on the top, and the focal fish that lose are at the bottom. And you can see that having a greater absolute aerobic scope advantage significantly predicts the ability of a focal fish to win a trial. In the middle graph, we see a weaker, non-significant version of the same trend when it comes to a maximum metabolic rate differential, and we see a weaker reversed version of that trend in the right graph with standard metabolic rate. And that actually biologically makes a lot of sense, right? Because if an individual has a high MMR, sure, that could help them win a fight, but sometimes those individuals will also happen to have a high SMR and thus a small scope. So that effect will be undercut. And the reverse is true with SMR. If you have a low SMR, that probably contributes to your ability to win a fight. But if you happen to also have a lower MMR, well, it's not going to be that helpful, right? So this supports not only what's been found in the literature regarding interspecific competition and absolute aerobic scope predicting it, it holds that specifically aerobic scope is the strongest predictor as opposed to other metabolic traits. But what's actually happening out on the piling? This is all cool, fun lab study, right? But is it representative of what's happening in the natural system? I hate PowerPoint sometimes. Give me 5 seconds. Here we go. So friends and I did a series of visual dive surveys on these concrete pilings. And here's what we found replicated across all 12 pilings. So first thing you should probably notice is that multifilus is by far the most abundant generally, and it doesn't show a wild depth trend. Aquapenis is not super abundant and doesn't change much with depth. Cristata is actually quite abundant right at the surface, in many cases more abundant than multifilus, and then it falls off ridiculously quickly with depth. Literally not a single cristata was found below 6 meters, right? And so if your first thought is of the classic Joe Connell ecology papers with the Balanus and Thamellus barnacles, do not be the first one to jump down that rabbit hole. We started exploring, are there any differences, or might there be any differences between multifilus and cristata that would support perhaps competitive exclusion upwards? So these are the fish-based distributions of these two species. Multifilus is exclusive to the northern Gulf of Mexico, and Scartella is all over the darn place. So that suggests that perhaps multifilus is a bit more of a specialist for this sort of environment, this sort of climate, whereas Scartella is a bit more of a generalist. And that would be consistent with this idea that multifilus, with its, I'll say, superior energetic setup, is better able to compete and push Scartella upwards. The missing piece of that framework, though, is that the upper part of the water column would need to be really not favorable territory. And you'd have to wonder why that is. We've got 2 ideas. First is these fish, as you may have seen in that photo earlier, love to sit in barnacles on the pier pilings. And the tide's actually pretty significant in the ship channel as well as, waves. So frequently you can see fish in barnacles exposed above the water line, and they don't look like they're having a fun time. So desiccation could be a serious challenge for fish that are trying to shelter in place near the surface. A second challenge that we didn't immediately think would be a huge issue is avian predation. This was taken by a lab member. This is an egret right in town here eating a very large Scartella. So anecdotal evidence, but it shows that it's at least a dynamic that's taking place here. So if this is really the mechanism that's supporting the structure here, this is consistent with these variable, invisible, innate metabolic traits predicting competitive ability, which in turn might be structuring the spatial layout of this community, which is really interesting and exciting. So would you believe the same dynamic plays out in my living room most evenings? Here we have Cricket using her superior competitive ability to monopolize this box on the cat tree, where she's better protected from not so much avian predators, but from me swooping by to pick up a cat to bother. And Nutmeg being competitively excluded upwards, where she has no choice but to let me pick her out and go sit on the couch. I'm happy to answer any questions about study or about using household pets to inspire animal behavior studies.
We still have enough time for questions.
I'm wondering if you went 1,000 generations into the future, the outcome of this aggressiveness of multifilus, would you see these habitats completely dominated by this species, or are there other life history offsets that allow the other 2 species to persist despite being, lower frequency or?
Really good question, and that's why I have a whole bunch of otoliths from these guys sitting in the lab upstairs that I have not analyzed yet. But you're right to ask that. It's very possible that there are various life history advantages that would, for example, allow Scartella to reproduce more rapidly and colonize and adapt to new pressures more rapidly than multifilus can. It's also possible that by having an elevated metabolic rate, multifilus reproduces more quickly, and it is purely a factor of geographic location and local population. And I also want to point out that this is, well, it's kind of sort of a natural system. It's also a man-made piling. So I'd be really interested to check out, is this dynamic even representative of what's on an oyster, for example? But yeah, no, these life history trade-offs are a very good question, and I'll get back to you next year.
So follow up to that.
Yeah.
What are you gonna do with the otoliths? Just look at growth or?
Yeah, so these fish, despite not being what the type of cryptodontic fishes that are most frequently studied, which is usually coral reef fishes, they have a fairly extensive pelagic larval phase compared to non-cryptid benthic fishes. It's a fairly large percentage of their lifespan. And so I would hope to delineate growth rates in between the different phases of, okay, well, first establish how long the pelagic larval duration is, and then try and establish a growth rate based on when they're larvae, when they're juvenile, when they're adult, because I imagine that would be the biggest predictor of how rapidly they can turn over. But the ultimate goal is a generation time estimate.
So when you were how did you assess the MMR? Did you do like a chase? Was that?
Yeah, no, I didn't have time to go into it, but yes, so it was a chase protocol. Had to pilot it out for each species, but it ended up being very comparable for all three. I placed them into a large chase bin and chased them by hand, reversing direction so that they didn't, to get used to it and ride the current. And my standard for when they were maximally exhausted and could go in the respirometer to measure MMR was that they lost their riding response, which for blennies that sit on the bottom is extra significant, I think, because they have the bottom to rely on. If they're falling over, they're really done.
Gotcha. And then what was you said that you assessed the aerobic scope for each individual.
Yes.
What was your time in between running those behavioral assays and assessing the aerobic scope?
So the aerobic scope assessment came first. We would capture them and we house them for minimum 2 weeks, and then did respirometry to get SMR and MMR aerobic scope. And then it was a minimum week and a half. I'm going to say 10 days was the exact amount before behavior.
And the purpose was to look at interspecific competition. In the field, did you see intraspecific competition or any aggression?
I mean, anecdotally, sure. Yeah, it's hard to see a little bit. I don't know if you've been diving at Ship Channel before, but the visibility is not clear. It's still fun though. But when you go down, yeah, you see plenty of all three species chasing each other constantly, passing around. So I didn't really look at that dynamic here because I wanted to look at the overall species distribution, but I'm sure there's a lot of interesting questions that could be asked with intraspecific competition in that system.
Okay, thank you. So our next speaker is Ethan Gass. His talk will be on characterization of Texas sheepshead highlights increased fishing factor on spawning aggregations.
Well, thank you. Yeah, so instead of blennies fighting each other, this would be a story of sheepshead loving each other. Yeah, I'm Ethan Getz. My co-authors on this one were Chaz Downey and Kat Eckert. We did a deep dive on sheepshead here. But first, I'll talk about spawning aggregations.
It's not that computer. The slide out one.
Okay, so fish spawning aggregations, this is when fish gather together in really large, dense schools to spawn. A lot of times they'll gather around like a reef promontory or a seamount or a jetty maybe. So it's a lot of economically valuable species. We've got these twin spot snapper here. That's a lot of other snapper and grouper species that do this. And problem arises when people get involved. Of course, when fish gather in large numbers, people are always interested and it's easy to exploit some of these species. Two really classic examples are the Nassau grouper and orange roughy. Folks have fished those heavily, exploited the spawning aggregations, and both species have experienced population declines because of it. We've got one of these species in our backyard, and that is the sheepshead. They're known for that gnarly set of chompers they've got. They eat a lot of crabs, crustaceans, barnacles, things like that. And they support some valuable fisheries, so both recreational and commercial fisheries throughout their range up along the Atlantic coast and throughout the Gulf. They spawn from February to April, kind of peaking in March, and they'll gather around jetties, nearshore reefs, artificial reefs, things like that. And there has been some evidence for overfishing sheepshead. We've seen reports way back in the 1800s off Louisiana. Folks exploited those populations and spawning aggregations and led to some population declines and reduced sizes of sheepshead in the area. So more recently, this study came out assessing the vulnerability of a bunch of species in the Gulf of Mexico. They looked at 28 species overall, and they ranked them on intrinsic vulnerability, which is related to life history. So species over here on this side are the ones that form spawning aggregations, have a tighter spawning window, And so they're more highly vulnerable compared to these species that are form less dense schools when they spawn. Extrinsic vulnerability is related to the fishery. So if there's a lot of protections in place or not a lot of harvest going on, there's lower vulnerability there. The species up top are the ones that have, let's say, higher quotas or easier to catch. And so to pick out a couple examples, you'll notice Goliath grouper and Nassau grouper down here at the bottom. We know that they form spawning aggregations, and so intrinsic vulnerability is really high. However, they're very protected throughout a lot of their range. In a lot of places, you can't harvest any of them. So that's why extrinsic vulnerability is so low. There's essentially no fishing pressure. Sheepshead, you'll notice, are way up at the top there. So they're kind of enduring the perfect storm of high intrinsic vulnerability and extrinsic vulnerability. They form dense spawning aggregations, and they're also fished commercially and recreationally, and bag limits aren't as strict. So knowing that, we wanted to take a look at sheepshead using Texas Parks and Wildlife Department's long-term monitoring data. So our objectives for this one were to look at a little bit of sheepshead ecology, look at distribution and abundance, characterize the fishery spatially and temporally, and then really hone in on the spawning season and evaluate targeted fishing pressure during that time. So to do that, we utilized our long-term gillnet data. So Parks and Wildlife every spring and fall goes out and they set gillnets throughout all the major bays in Texas. For this project, we looked at data from 1983 to 2023, and that represented about 30,000 gillnet samples total. So a massive data set there. And then we also used data from our angler creel surveys. So again, all up and down the Texas coast, we'll head out to boat ramps, and as anglers are coming back in from day of fishing, we give them a series of interview questions, asking things like what they were using for bait, how long they were out there, what they were targeting, and then we'll count and measure the fish that they brought in. So again, looking at 1983 to 2023 for those data. Analyses for this project were pretty straightforward. We looked at distribution, generated some heat maps, looked at abundance trends in those gillnets, and then used boosted regression trees to highlight some environmental predictors of sheepshead abundance. And then using the harvest data, we looked at catch rates, angler preferences, and seasonality of harvest. So here's a look at sheepshead distribution based on our gillnet catches. You'll notice catches are a little bit lower in the upper Ghost, but south of Galveston Bay, we see high catches around Port O'Connor, Port Aransas, down around Port Isabel. And the other thing you'll notice is that catches or higher catches of sheepshead are associated with passes. So if you look at the secondary and tertiary bays, Baffin, Nueces, Copano Bay, we're not getting a lot of sheepshead back there, but right around the jetties in Brazos Santiago Pass, up here in Port Aransas, Port O'Connor, that's where we're seeing most of the sheepshead kind of associated with Gulf passes. Then looking at sheepshead abundance, so again, this is from our gillnet catches. This is annual catch rate over the entire time series. And there's not really a trend throughout the entire time series. But if we look over here at the most recent years, we've got some decreased catch rates. And actually 2023 was our second lowest year of the whole time series. So something we might want to keep an eye on as managers there. This is the output of the boosted regression tree analysis. So that highlighted salinity and temperature as the two most important environmental factors influencing sheepshead. Salinity, you'll see they like almost just about full strength seawater. Their abundance seems to kind of peak right around 30. And then temperature, they seem to prefer moderate temperatures in that 20 to 25 range is where we saw a lot of the fish caught. One interesting result we got here was that in the literature, the previous maximum salinity tolerance for sheepshead was around 60. And here we were actually observed a few sheepshead up over 70 PSU. So they seem to be a little more salt tolerant than we previously thought. And then this figure, I think, really tells the story. So the gray bars here is the percent of sheepshead harvest per month. So that's looking at the angler creel survey data. And you can see that the majority of sheepshead that we see come into the boat ramps were harvested in March. And so that's right during the sheepshead peak spawning time. The dotted line there is angler catch rate, so catch per angler hour. And again, you'll see that really peaks during the spawning season in March. So not only are people harvesting more then, but they're more efficient in doing it. They're spending less time on the water to catch more sheepshead during that time. Here we're looking at species sought data. So this is when if an angler brings back some fish, they've got a sheepshead, and we've asked them, what were you fishing for today? And they tell us either we were primarily fishing for sheepshead, or we've just lumped everything else as other. So maybe they were fishing for redfish or trout, something else. You can see during the warmer months, kind of May through October, the vast majority of people that brought in a sheepshead were not fishing for a sheepshead. They were targeting something else, and the sheepshead was incidentally caught, and they just kept it and took it home. That trend flips during the spawning season. So again, peaking in March, the people that brought back sheepshead were directly fishing for sheepshead, and that's what they got. So it seems to be a targeted fishery during the spawning season, and then just kind of incidental take throughout the rest of the year. And then not only was sheepshead catch temporally kind of concentrated, but we've also got some key areas along the coast where harvest is taking place. And it's right in the backyard here. It's right around Aransas Pass, Port Aransas, Corpus Christi Bay, the ship channel. The portage eddies right here are really locally known as a great spot to catch sheepshead. And so that's where we see up to, in some years, it was over 70% of the coastwide harvest was right here in this area. Other important areas were up in Port O'Connor and a little bit up around Galveston as well. And then this was kind of an interesting trend too. This is the percent of sheepshead harvested during the spawn each year. So if you look at a year's worth of sheepshead catches, how many of those were caught during spawning season? And so in the beginning of the time series, it was relatively low and kind of increased up until the early to mid 2000s, when we had most of the sheepshead caught in a given year were caught during the spawning season. Since then, it's kind of plateaued and even come down a little bit for some reason. Hard to kind of say why. We've got some ideas, but it would really just be speculation. One interesting trend though is again looking at our gillnet catches. Since around 2012, 2013, those catches have declined, so sheepshead abundance has declined. And that's right around the same time period that we're seeing this percent harvest during the spawn kind of plateau and maybe dip down a little bit too. So maybe related to angler pressure or sheepshead abundance overall, but it's kind of difficult to say. I think certainly what we can say from these results though is that sheepshead experienced increased fishing pressure during their spawning season. So this is a picture of Packery Channel during the spring. And you can see all those people are gathered around to catch sheepshead, and that's just a typical day during the month of March. So there's certainly increased pressure on sheepshead in Texas during the spawn. And something that as managers we might want to pay attention to is that as of now, sheepshead are not the most sought after species. A lot of folks would prefer, spotted sea trout and flounder and some others. But in the recent years, we've implemented more restrictive regulations on some of those species. And so if folks are going out and saying, okay, I can't keep as many trout or flounder, for instance, maybe they'll switch their attention to sheepshead, and they certainly know how to do it. So that's something that we may want to think about in the future. Some other things here, I think this study really highlighted how useful it can be to couple the fisheries independent data with the fisheries dependent data. So using both our gillnet catches and the angler harvest catches, coupling that data to kind of tell a full story here helped a lot. And that's something we do regularly that helps. So right now we've got a minimum size limit on sheepshead of 15 inches. And that was put in place to allow sheepshead to grow to a size where they can spawn once and then become susceptible to harvest. So their first year of spawning, they're protected. Another thing I think these results highlight is it'd be interesting to look more at movement and habitat use of sheepshead and certainly kind of define what constitutes essential spawning habitat, what structures exactly they're using to spawn. And then again, managers, I think, could be thinking more about taking life history into account when we make decisions. So right now, throughout the Gulf of Mexico, at just about every level, there's not a lot of fisheries management plans that take into account spawning behavior and some of these life history traits. So again, something to think about in the future. So with that, I'll take any questions. Yeah, Jerry.
Yeah, thanks for the talk. Have you looked at the offshore population? Or have the capacity to look at the offshore population, how they compare to the inshore population?
Not specifically for this project. We do some trawling offshore, which I'm not sure. I would imagine we probably don't pick up a lot of juvenile sheep's head out there because they do recruit to the bay and seagrass meadows specifically. So yeah, not much there. We do have some data from our artificial reef surveys. And I know that on those surveys, you do see increased sheep's head populations or densities on the artificial reefs during the spawning season as well. So they're certainly using those artificial reefs to gather around and spawn.
Any thoughts on if there's been shifting seasonality in spawning aggregations? I mean, you kind of showed where harvest was occurring during, I'm not sure what you defined as the spawning season, but any thoughts on if that shifted at all? We've seen that in a lot of other species. I'm wondering if that's happened there, and that could describe why you're seeing fewer harvested during a certain season, if that season shifted across.
Yeah, so we define the spawning season as February through April. And we looked into that a little bit and didn't see a ton of evidence. So I think, yeah, something to keep an eye on for sure, but I'm not sure it's telling the full story here. Yeah.
Do you suspect that most of the catch is occurring in those passes as sheep's head are moving offshore? Or do you think that there's actually people targeting offshore locations where there's spawning aggregations of sheep's head? Or is it neither of those things?
Yeah, so we were just talking about that picture, that Packery Channel, the people that know that area, that's not exactly right on the jetties. That's, kind of right as you cross the road, there's a deep pool right there where those people are fishing. And then you go out to the jetties and you'll see big spawning schools out off the jetties, but we're not really sure why so many people target right here, whether this is just an area that's kind of a bottleneck that sheep's head have to move through to get offshore, or whether they're actually aggregating to spawn right there. So that's why I was thinking kind of habitat use would be interesting to look at in the future. If you go out to the nearshore reefs and artificial reefs in the springtime, they're loaded with sheep's heads. So they're certainly using those structures, but it's hard to say whether this is kind of a stop off point or whether they're spawning right there too.
Thank you, Ethan, for
Yeah,
sharing
thank you.
with us. Next, let's welcome Judy Matthews. Her talk will be using metabolic traits to grow in the Western Gulf of Mexico.
So as you said, my name is Isabel Cummings and I'm with the Fisheries Parks and Wildlife Department. And here I'm presenting some research to you done off of previous data titled The Environmental Influences on Juvenile Atlantic Croaker Growth in the Western Gulf of Mexico, or specifically Texas. So I'm sure most of you are already pretty familiar with an Atlantic croaker in general, but just to give some species background to give you an introduction to this project. Atlantic croaker is a marine finfish species found commonly throughout the Gulf of Mexico and even as far up north on the other side of the coastline, as all the way almost up to Maine. And they are a marine finfish from the family Cyanidae. So if you're not familiar with Atlantic croaker, you're probably familiar with other members of their family, such as red drum or spotted sea trout. Now, Atlantic croaker typically spawn in the fall seasons, so they will move offshore in aggregations to spawn, and their larvae will be transported back into low salinity estuarine habitats for them to begin to grow and take advantage of the high productivity of spring in late winter and early spring months. So Atlantic croaker are a very important bait fish. They are increasing in popularity both commercially and recreationally. And particularly in Texas, they have seen a lot of popularity as a bait fish, but they are also commonly known to be a food fish as well. So they offer both economic and ecological importances throughout their entire distribution. They have even for some time were a major component of the groundfish fishery in the 1960s. However, they've consisted of so much of this groundfish fishery, which was basically used by companies to produce cat food and use other bait fishes for similar objectives. And they did suffer a pretty notable decline at the time. However, currently populations are much more stable. Now, despite their popularity as a commercial and recreational species, there is unfortunately limited knowledge on important life history events that are necessary for effective fisheries management. Particularly age and growth. So there's been a lot of publications on adult croaker ranging from age of 0 to up to their maximum age of around age 8. Although most Atlantic croaker don't typically get older than about 2 years old and they reach sexual maturity at age 1. There have also been some previous studies done on larval and early juvenile stage Atlantic croaker. However, these typically cut off around 80 days old and they don't have too much information from 80 days on until that first year of maturity. So there is unfortunately limited age and growth studies for young of the year juveniles. So young of the year meaning fish born that year, age 0 fish. There's been even less known age and growth on juvenile Atlantic croaker, specifically in the Western Gulf of Mexico, and for the sake of this project, Texas. And there's not a lot of specifics looking at whether larval, juvenile, or adult on how salinity or other water quality parameters impact Atlantic croaker growth. There's even been some conflicting studies looking at salinity effects on juvenile and larval growth. There were some studies in 1999 and through 2016 that suggested that lower salinities were more conducive for growth for juvenile and larval Atlantic croaker alike. However, there was a study published in 2021 that suggests that higher salinities may actually increase the rate of growth after a certain age point. So the previous study suggesting that low salinities were better for faster growth, we're looking at fish younger than 80 days, so not typically larger than a certain size, whereas this 2021 study was looking at salinity over a multitude of different sizes, and they found that salinity had a negative, or had higher salinities were worse for growth for smaller fish, but for as you started to reach these larger sizes and Atlantic croaker became more tolerant to higher salinities, they actually saw increased growth in high salinity fish over a size threshold of 71 millimeters. Water temperature has also been considered to be an important factor when it comes to growth, particularly in cyanids. Other studies looking at red drum and sea trout have shown that warmer waters are typically help with faster growth in juvenile and larval cyanids. However, there has not been a lot of research published specifically for juvenile Atlantic croaker looking at temperature. So that provides you a good background of the literature, which sets the stage for the objectives of this particular project. So we have 3 key objectives that we've determined for this project, with the first one being to evaluate different growth models for monthly young of the year juvenile Atlantic croaker growth using length frequencies from our Texas Parks and Wildlife routine sampling data. Our second objective was to compare growth trend variations using these models along the entirety of the Texas coast, looking at differences in major bays. And our last objective was to determine environmental impacts on monthly growth with an emphasis on salinity, since that seems to be the dominant factor influencing Atlantic croaker growth in previous literature. So moving into our methods, as I mentioned, that we did use the Texas Parks and Wildlife routine sampling data from 1990 to 2023. We took both bag seine and bay trawl data to help give us a good amount of length frequencies for these juvenile Atlantic croaker. And for the sake of interpretation, we separated our major bays into 3 different regions, with the first region being titled the Upper Coast region, including Sabine Lake, Galveston Bay, and East Matagorda Bay. We also have our Middle Coast region, which includes West Matagorda Bay, San Antonio Bay, and Aransas Bay, and then our Lower Coast region, which is Corpus Christi and both Upper and Lower Laguna Madre. So from this massive data set, we were able to extract the fish that fit the size ranges for young of the year Atlantic croaker, and we extracted over 435,000 individual fish for this project. So this graph right here is just showing the total length of each fish, the mean total length of all the fish that we looked at per month, which was roughly what we used for age, considering their spawning season time. And we modeled growth using multiple different growth models to avoid any bias from just using what's been done previously. So we use the von Bertalanffy model, the Gompertz model, logistic, Richards, and power growth curves to try to figure out the growth curves for the species. And to look at water quality parameters significance, we use a generalized additive model. So alongside the fish collected, Texas Parks does collect environmental data, including salinity, water temperature, turbidity, and dissolved oxygen. So we looked at all of those variables in our model as well. So for our growth models, we selected Richard's model based on it having the lowest AICC and best overall fit to our data. And we think that Richard's model might be a better fit than the previously used Gompertz and von Bertalanffy models for modeling juvenile growth, because it has an additional parameter that allows it more flexibility to help compensate for any changes in growth rate that is typically characteristic of young fish, since they grow a lot during the first few months and then they tend to slow down a little bit. So this equation at the bottom is just our coast-wide growth curve equation for young of the year Atlantic croaker. But we did take the Richards model and separate it out into our 3 different regions. So we have our upper coast regions graphed in green, our middle coast in red, and our lower coast in blue. And as you can see, there is a pretty predominant trend occurring in this data set, with lower coast fish tending to reach their maximum size much quicker and have a much steeper curve than a middle coast and upper coast. So we actually saw, when looking at individual bay systems as well, that there is a pretty reasonable north to south trend of increasing growth. So the further you go from Sabine Lake all the way down to Corpus Christi and Laguna Madre, Atlantic croakers seem to reach larger sizes faster than further north. So I'm sure most of you are familiar with the water quality compositions of our bays, but just to touch on some things that are pretty commonly noted about the water quality, I wanted to put up this graph that is showing mean annual dissolved oxygen, turbidity, salinity, and temperature for our upper coast over here, middle coast, and then lower coast all the way on y'all's right. So moving from north to south, our growth of our Atlantic growth rate increases, our turbidity decreases, our salinity increases, and our temperature increases as well. So to further parse out how these were significant, or if they were significant in the model, we included these, and as I mentioned, our generalized additive model, and we determined that only dissolved oxygen, salinity, and month were significant for predicting rate of juvenile croaker growth. This graph right here is showing annual mean salinity on y'all's left, and on the right is the annual mean dissolved oxygen compared with our annual mean growth from our Atlantic croaker. So you can see pretty consistent trends of as mean salinity increases, growth rate tends to increase, and as annual mean dissolved oxygen decreases, annual mean growth also tends to decrease, or yes. So we think that dissolved oxygen might be significant in the model due to its relationship with temperature. We do see that increase in temperature as we move from north to south, so that may explain the decrease in dissolved oxygen, as there hasn't been a lot of previous literature suggesting why dissolved oxygen may be important. However, salinity, we do see that distinct gradient moving from north to south, coinciding with the gradient of increased growth. So that suggests that higher salinity may actually equate to faster growth, which is surprising considering the amount of previous literature looking at larval and early juvenile Atlantic croaker growth being actually inhibited when exposed to these higher salinities. But there was that one study in 2021 that proposed that fish above a certain size threshold, and that study was 71 millimeter standard length, actually began to benefit from higher salinities and have faster growth. And most fish in this study were above that minimum size. And to just kind of break that down further, that minimum size is this black line on the graph that was proposed in the 2021 study. We have total length on the axis here per each month. And as you can see in the first few months, the mean total length for each of the regions are highly similar. But once we start getting above the 71 millimeter threshold, you'll really begin to see lower coast pulling ahead in growth. So this does further suggest that salinity is playing a factor, but specifically after once they've reached a certain size to withstand the salinity and may be able to redirect energy from osmoregulation into growth instead. So to further break this down, I have a growth per month Separated on the axis again for the upper coast in green, middle coast in red, and lower coast in blue. And we do see these distinct regional trends moving from north to south alongside salinity changes. And that brings us to ask why we might be seeing these differences in salinity per bay. So why might there be a benefit for these larger sizes to occur earlier on in life in higher salinity regions than in lower salinity regions? So this may be a possibility to reaching these growth early may be a possibility to help withstand higher salinities faster, so they can reach those size tolerant sizes faster than what would be needed in lower salinity regions. It may also allow them to take advantage of spring productivity. So there is a lot of greater prey availability at the time. They may have competition with other species in these bay systems. And as they typically start out in low salinity estuarine habitats and move outwards into bays to diversify their prey as they get older, there may be a benefit to reach those sizes earlier and higher salinities to move outwards and take advantage of that prey before other species are able to do so. So to summarize the project as a whole, here are our three main objectives again. We wanted to evaluate different growth models for monthly young of the year growth using length frequencies. And what we found was that Richard's model was the best fit for young of the year growth, primarily or most likely because it has those additional parameters that allow for the flexibility to model such quick changes in growth throughout their first year of life. We also looked at our second objective, which was to compare growth trend variations along the Texas coast. And we determined that there was significant regional variation in growth from north to south. And our last objective was to determine environmental impacts on monthly growth with emphasis on salinity. And while we didn't see temperature to be significant, we did determine that salinity exhibits a size-dependent effect on growth. So that moves us into the conclusions of this project. The goal of this project was to look at a data gap between those early stage juveniles that have been studied and those adult fish that have been studied, and try to look into the later stage juvenile fish aging growth to provide previously unknown data for Atlantic croaker along the Texas coastline. And what we saw was that juvenile growth typically does exhibit distinct regional and environmental patterns, changing as we go from northern bays down to southern bays, which may be representative of growth patterns throughout their entire distribution. So what we're seeing on this Texas coastline may also be visible on in other states and throughout the rest of their distribution as well. But unfortunately, because there's not a lot of literature on that. So, and lastly, this research establishes an important baseline information for fisheries management as environments continue to change. As we know, waters are warming, habitats tend to be degrading depending on where you are, and there's a lot of change going on in these estuarine habitats. So as these changes occur, it is important for us to understand how water quality parameters impact species such as Atlantic croaker, so that we are able to manage the situation and be able to protect them as things continue to change, so that we can continue to use these fish as a resource for generations. And lastly, I would like to thank all of the Texas Parks and Wildlife people for going out and performing all this research, for pulling the bag seines and bait trawls, and being able to collect this data, this impressive data set over such a long period of time. I would also like to thank Joel Anderson, my co-author on this project, for help bringing this idea to my attention. And lastly, I would like to thank all of you very much for listening and open up the floor for any questions.
Any questions?
Yes.
Is there evidence of earlier recruitment in the lower bays? You know, near.
I have not seen, but if you do look at we did filter our length frequency data pretty heavily, because in those later months, like months 11 and 12, you would start to see fish begin to enter that are much smaller sizes, so the next year's cohort. And I did notice a trend that in those lower bays that there were some instances, like there was a greater amount of smaller individuals arriving on, but I don't know if that's conclusive enough to be able to suggest that is a full-on trend throughout the entire distribution. Yes.
Did you see any differences in like abundances or catch rates along the coast?
Yes, when it comes to catch rates, they are typically caught most often in, I believe, Corpus Christi, Upper Laguna Madre, not so much up north in like Sabine Lake, San Antonio Bay. But I didn't look too extensively into that.
Thank you.
Thank you very much.
Stocked fishes, a case study with a white bass. Wait.
Thank you.
Hello, everybody. I'll start talking about the white sea bass. That is the Atractation nobili. That is the largest cyanid species in the Pacific coast of North America. It can reach 1.7 meters on total length. That means can be taller than me. And it can weigh 43 kilograms. This species is distribution is in the northern California to the south in the Baja California, Mexico, including the Gulf of California. And during warm season, like El Nino, it can be found in Alaska. So that is interesting. And because the quality of the beach, it is an important species for the commercial and recreational fisheries in both countries or through the distribution. As historical records in the fisheries or the catch, it has been, I don't know, this is a species or a fishery very old. It started before the 1950. And as a summary, you can look in here, then from the 1915, and it has a very important decrease in the landing in the 1820. So after that, and based on the low abundances or catches, then in the 1982, the Hub Seaworld Research Institute start a restocking program. So it has around 40 years. And during the same period, they start some fisheries management restrictions as a closed season, the gilded band, and also in the sport fishing, they start reducing the number of fish that people can catch during one-day trip. So after that, and now there have been released around 2.7 million hatchery juvenile fishes in the Southern California Bight. And as a strategy of this program, the juvenile fish has been released in different areas in all the bay. So to be sure the fish are, well, with a good distribution in all the area, including the Catalina Island. So before release, each juvenile has been tagged with a code wire tag. You can look an example, and this is the size of the tag. So when they recapture the fish and read the tag, we can know all the record of the hatchery as the spawning date and the temperature and salinity records of the water from the tank, of course. And well, now there is a pressing need to estimate the contribution of the stocking program of the fishery, which require accurate identification of the restock individuals. So there is another programs, including here in the Gulf of Mexico. So another studies to use alphabet trace element to identify the hatchery fish. And some of them has been successful, as for example, the red snapper here in the Gulf of Mexico, but fewer use stable isotopes. And for the carbon, we use oxygen and carbon isotopes because we can infer environmental condition of the fish when they has been raised. In the case of delta oxygen 18, we can know about the temperature of the formation, and something important is to know, and we have temperature and the relationship between temperature and the oxygen. So if we have a change of 5 degrees in the environmental temperature, we can look at variation in the oxygen 18 of 1 part per mil. So 5 means a lot. In the case of the carbon 13, we can have another kind of information because that reflects the information or different sources as metabolism, dissolved in organic carbon and food sources. So the main factors controlling these values are age, habitat, temperature, differences, or when the fish change in trophic levels, and the habitat. So the question is, based on this restocking program, if we can use the stable isotope as a tracer to identify the white sea bass from the hatchery released in the Southern California Bight. So for that, we have different group of fishes depending on the stage. So we have the juvenile stages. Those fish has been spent whole life under hatchery conditions. They are small, they has never been released. And we have another group from adults and subadults. And they have been released when they were juvenile in the Southern California Bight and then recaptured. This program has different small programs to recover the head of the fishes. And once recovered, all the heads, as you can see in here, they read the tag and they know which fishes has been recovered. And we have another group of fish from wild, and we took them from the commercial and recreational fisheries from the Southern California Bight and the northern part of the Baja. So once in the lab, we took one otolith and it has been including resin. We did a transversal cut and we just look the core that it is here. And with the micro drill, we just take the carbonate from the core and we take samples weighing about 50 and 100 micrograms. Those were sent into the Arizona lab to analyze, and we have in total 119 otolith samples. So also for the wild fish, we read the age, reading the rings. We estimate the age, reading the rings from the core to the age of the otolith. So we have fishes from 0 to 10 years old, and because we know the age, the year when they has been cut, we can back calculate the spawning years. And the spawning years was between 2015 and 2022. And for the hatchery fishes, we have ages from 0 to 20 years old and spawning years from 1997 to 2024. The 2024 are fishes, the juveniles. So as a result, I'm gonna show you at first just the results or the isotopic composition of the hatchery fishes. So we have the carbon and the oxygen, and we have three groups: the juveniles from hatchery, subadults, and adults of the recapture. And you can see a high variability in all the information. So the question at this point, it was, could this be reflected the interannual variation? Because we just see a lot of variability in the carbon and differences in the oxygen. So for that, trying to understand these values, I'm using the same isotopic values, but now I'm grouping as a function of spawning years. And we have two groups based on the carbon results. And we have, in the oxygen, we don't have that much variation, but this is something very different in both isotopes. So trying to understand those values, we get back to the hatchery or the tank temperature and salinity. And I'm showing here the spawning years of each fish and the temperature and salinity. So we have differences before 2015. We have a high variability in the temperature, and from 2015 until now, we have the same temperature in all tanks, around 18 degrees. So I'm showing you here the same with the salinity, and the fishes with temperature around 18. Now the tank has been spent like at 34 units of salinity. So that is important for us because with that, we can suggest that the breeding condition of hatchery fish differ from those when we are thinking in the 2015 and before. And also, the low values of the oxygen 18 reflect higher reading temperatures. And in the case of the carbon, we will have, maybe we have different diet sources. So with that, for a greater accuracy, it's, we will focus our analysis only in those fishes has been spent during or after the 2015 because the hatchery conditions. So now here I'm comparing, we have the hatchery data, all the stages in gray, and now I'm adding the wild fish from the Southern California and Mexico. And with this, you can see, if we just look the oxygen, we can say the oxygen is not a good tracer to discriminate between groups. But, and you can see it here too, in the top chart. And also, we have, in just looking the carbon, we can have, we can see or say that is a good tracer to identify between wild and hatchery fishes. And we can see the same result or the same pattern if we compare between different life stages. So the oxygen, it is no differences, but we can see the overlap between recaptured individuals with the hatchery juveniles. And we have the clear differences between wild fishes from Southern California and Mexico. And also you can see high variation in the carbonate values in the fishes from Mexico. So we don't know yet what that means, but something else. So as a summary, we can say the carbon is a good tracer to identify the hatchery fishes. And this approach can be successfully discriminate both groups of fishes as long as similar temperature and salinity are maintained over the time in the tanks. And ongoing research with complementary identification techniques seeks to estimate the hatchery contribution of the fishery. And that's it.
I said it was. And you thought that was the way they do it?
Okay, everyone, welcome to come back. We still have 3 more talks to conclude the morning session. 2 of them are from Fisheries, and there is another one, or only one, from the vertebrate science. And the first, let's welcome Annie Taylor. Her talk will be on developing a method to measure current flow around the artificial reef.
Okay, thank you for the introduction. My name is Annie Zeiler, and today I will be presenting to you a portion of my thesis work in which I'm developing a method to measure current flows around an artificial reef using a Sontec M9 EDCP. So a little bit of background on artificial reefs. These are structures that are placed in areas to increase fish habitat. They attract reef-associated fish. So they're highly targeted in fisheries and are a big focus for additional habitat. We know that reefs alter current flow regimes. However, there are some common problems that occur with this, such as scouring and burial. This affects the longevity of these reef structures, as well as reduces the vertical relief, impacting fish and other marine organisms. Here is just a quick video of one of our structures at the RGB Reef. This is known as Biggs Pile. As you can see, it is just literally a big pile of concrete railroad ties. But there's a lot of marine life on it. We have tons of fish as well as other encrusting organisms and marine organisms that settle on this reef. So we're interested in currents. These reef areas combined with these currents create a unique ecosystem. It creates physical and biological changes as these currents are bringing nutrients to this reef area. Scouring also occurs as a result of these currents and can be observed to be 3 times larger than the structure itself. So the main inspiration behind this project actually comes from this image. This is some side scan imagery of an oil platform, artificial reef. And as you can see, there's a ton of fish associated with this reef, which have been observed from 60 meters away. And so we want to know, why are there fish here? Are there eddies? Are there lower velocities occurring here? Is it easier for these fish to get food and possibly not have to fight the current? And we want to know, how can we measure this? And why even measure currents at all? There is a lack of data on current measurements on reefs currently, especially artificial reefs. Reefs are expensive to deploy, so knowledge of these flow fields around these structures could inform future reef deployments and design, so that we can maximize the benefits of these reef areas. And this might not only apply just to artificial reefs, but also possibly oyster beds or other inshore habitats. And then current velocity measurements could be used to examine additional factors that influence scouring and fish utilization of these artificial reef structures. How do we even measure currents? There are plenty of tools available to do so, such as acoustic Doppler current profilers, or ADCPs, acoustic Doppler velocity meters, flow meters, and tracer dyes. However, for this study, we are looking at measuring currents throughout the entire water column, so an ADCP is likely the best choice. And then why a Sontec M9 ADCP? There are other ADCPs that are available, and large-scale measurements offshore have been made, but these are in large areas with high flow velocities. There are also uplifting ADCPs, and these have temporal variation, but we can't see the spatial variation with these unless we had like 100 of them, but we don't. And so these require stationary deployment, so we would have to constantly move them around. So the Sontec M9 ADCP is mobile. With this, we can observe flows around multiple reef structures in one day. We can observe small-scale, low current velocities, and we want to know, can it be adapted for use in an offshore environment? So can it measure currents? Absolutely. These are used usually in rivers to get volumetric calculations. However, it has the ability to measure 9-bin increments throughout the entire water column. So in each one of these pixels, we're able to get speed, direction, GPS coordinates, signal to noise ratio, heading, as well as some other metrics, and we're getting data once per second. The M9 has the potential to measure these fine-scale currents around reefs. However, there are no refined tools or software to look at it this way. And the data then must be clean, filtered, and represented by vectors. So my overall objective is to develop a methodology to measure these flow regimes in relation to scouring and fish positioning around an artificial reef using a Sontec M9 ADCP. Onto our study site, which you may have heard of before, is the Rio Grande Valley Artificial Reef. This is the largest artificial reef off of the Texas coast. It's located 13 miles northeast of the South Padre Island jetties, and it covers over 16 acres of Texas state waters. The reef consists of sites of varying size and material, as well as replicates of these structure types. And we also know that this reef provides habitat, food, and protection for all life stages of various reef fish species, such as these red snapper here. To look at this whole reef, I created a 3D map using swath symmetry. However, for this portion of my study right now, we're focusing on a structure known as Biggs Pile. This is a pile of 6,000 tons of concrete railroad ties, and it's currently 11 meters tall. And then this is what Biggs Pile looks like as a diver is descending down on it. Onto my methodology. First, before we could take the Sontec M9 offshore, we needed to adapt or upgrade it with an RTK GPS because normally this ADCP is used in in-stream flows, inshore and in freshwater environments. So once we upgraded that, we use a point perfect correction service using a cell phone booster since this is an online server and we're offshore. And we can get corrections up to 12 miles offshore. And with this RTK GPS, we're able to get 2 to 3 centimeter accuracy in our GPS coordinates, giving us better accuracy for our current measurements. For our data collection, we started off trying 3 different methods. First, we mounted the ADCP to the side of our boat on a pole. And then we used the Sontec hydro board, which is up there in that corner. First, we used it on a stationary line where we would pull it to different positions and let it record for a few minutes and then pull it down the line. And then we tried towing it from behind the boat. And this ended up giving us the most stable and clear readings, so we stuck with that. We also use side scan sonar to measure fish abundance around these structures. And this this occurred immediately after our ADCP data collection and is then compared with our current data. Onto our data processing. Initially, we use a software called HiPack. However, this has limited functionality. We're able to get these great images, but we can't make any adjustments to our data. So used in conjunction with side scan, we can get these visual representations where we can see areas with velocities forming, and there's fish exactly where we would expect them to be. Since we can't make adjustments in this data, we had to move to a program that could correct, smooth, and visualize this data better, which is MATLAB. Before we can move into MATLAB, we had to use this Sontec software called RSQ, in which we would input our sound speed data from our Castaway CTV. We would clean the data for individual beam spikes caused by fish or structures. So likely this spike here is caused by a fish, probably at the surface. And it also happens if we go over structure with just one of the beams. So we can clean that up. And once that's all good, we can export each of the cells with the associated data into MATLAB. So I got to learn how to code. MATLAB's really fun. We started our script off where we can remove sections of bad data that are created by fish interference. We can then smooth our GPS speed spikes. So the blue line is our original boat speed. As you can see, it just like jumps up and down. By applying a filter, I was able to smooth that out and then put that back into our data, and it made our velocities look a lot better. We can then average this data by distance and then visualize these vectors over our 3D map, either by depth or by transect. Just briefly to touch on some of our results so far, we are able to visualize these currents around the RGB reef, and we have been able to see eddies form around Biggs Pile using HiPack, like the image I showed before. And we can also see deviations in currents observed near these structures. So this is a boat. It's kind of hard to tell, but here we're getting higher current velocities and then they start to slow down. So that's just one example of that. And then we are also able to see changes of current velocities and directions by depth around these structures. So these are 2 adjacent cells, cell 8 and cell 9, these are closest to the bottom. And as you can see, there are slight changes in these current velocities. So we are able to see that. On to my discussion, some of the challenges we've been facing so far. All of these softwares are like incompatible with each other. So HiPack works great for visualization, but we can't manipulate the data. The Sontec software RSQ can make corrections, but then we can't export those into other programs. And MATLAB, which we've decided to use, allows for these corrections, but the coding requires a steep learning curve. And then we're still working to validate these measured current velocities and directions using replicated transects. So if we go over the same structure a few times in the same day, are we seeing the same current speeds and current directions? Another way to validate this is with point measurements using ADVs and divers, and then comparing that to our Sontec M9 data and see if we're getting similar measurements. So in conclusion, we can use this M9 to record velocities around these reef structures. Our best results so far have been accomplished with MATLAB, since we can visualize these flow fields around the artificial reef structures at multiple depths, and this program then allows us to correct for the vessel speed. Some of the questions I'm still working on are what distance do we average these transects to get the best representation of these velocities around these structures? We're also looking into what is the best transect pattern to use over these reef structures, whether that be a grid pattern or possibly an X. We're also interested in figuring out the maximum sea conditions that M9 can be used in, because currently we're going offshore in only 1 foot seas, so that's like once every 3 months. And then what other types of habitat can we adapt this methodology for? Can we use this in oyster reefs or possibly inshore? And with that, I'd like to thank all of the Climb Lab members for helping me offshore, and as well as TPWD and Friends of RGB Reef for my funding. And with that, I will take any questions. Yeah.
I have two questions. First one's real quick. What is the maximum sea state you've used so far? Like, have you done it pretty calm days?
Yeah, we usually go out in like 1 to 2 foot seas. But I think we've gone out in like 3 foot seas, and the data just gets really jumbled.
My second question, sorry about being greedy. Do you anticipate having some kind of product from this that you can share, like some kind of 3D layer of current velocities and directions or something like that? Is that
That's the
something?
goal. We're struggling getting the 3D map like actually in 3D into MATLAB right now.
Yeah.
Still working on it. But these vectors are in 3D.
Yeah.
We just need to figure out how to combine that with our 3D map, so we can place them over that.
Thank you.
So I guess a little bit similar to what Terry said. So you're obviously only able to go out and do this on days that are like particularly calm.
Yeah.
So how do you capture that variability in velocities and things like that might shift as weather and, faster currents and things like that shift?
Well, we do have a seasonality bias for sure, since we are going off in flatter times. But with this new RTK GPS, I think we're gonna try to go out in a little higher seas so that we can see those faster current velocities and the changes that occur.
Do you have on that board that you tow, the board that you tow behind the vessel, do you have a motion reference unit at all? It's accounting for kind of heave or roll or any kind of
I
that?
think there's an internal one in our,
Roll and pitch, but heave is not something that ADCP is typically applying, at least with Sontec. Wish they did.
Thank you.
So our next speaker is
Going to start back up again with our fish and fisheries talk. Speaker here is Daniel Kemp, and his title is Morphological, Physiological, and Behavioral Biomarkers for Enhancing Southern Flounder Larviculture.
Hey everyone, so I'm Daniel here from Galveston, and talking about my work with flounder larviculture today, particularly with biomarkers for predicting their health and their post-release. So, start off, what are southern flounder? Why do we care? So these guys are a large flatfish. They are one of the few large predatory fish that lives in the estuary and salt marsh ecosystems in the Gulf of Mexico, as well as on the US, southeast, where they are of pretty huge economic and cultural importance, bringing in $1 million in Texas alone each year, as well as being a pretty huge part of the local fishing culture. I'm sure those of you that are in the scene are quite aware of the annual flounder run. However, these guys are not doing too hot in the wild right now. Based on these wild catch rates, we're seeing those populations decline pretty gradually, about 2 to 3% annually, since the late 90s. This is despite repeated restrictions on fisheries, including multiple seasons, tender bag limits, size limits, etc. And it's compounded by the fact that females are larger than the males and far more likely to be harvested. They're going to be more likely to hit that 15-inch minimum size to be legally harvested. So, to address this decline, we're seeing some great stock enhancement efforts from hatcheries run by TPWD in Lake Jackson, Corpus Christi, and Palacios. Those former two, Lake Jackson and Corpus Christi, are where my study was conducted, where they're using strip spawning to produce these larvae, However, these guys are pretty sensitive to both temperature and settlement space, among other things. So just like all flatfish, these guys are going to be metamorphosing right around 40 to 50 days old to that more flattened out adult stage, where they're only really going to be using the sides and bottom of the tank when they settle. So they won't use the full volume there. So we're seeing pretty high variability in their survival across those hatcheries. To address that, I'm looking to try and find some kind of early biomarker that can be measured very early on in their development. That's going to be predictive of their overall growth and health in terms of both morphological, physiological, behavioral endpoints. So my sampling for the process took basically 3 stages over the course of each sampling day. So I was going to both hatcheries at Corpus Christi and Lake Jackson to document and record the physiological parameters of these fish for 2 different local hatcheries. One in every 5 days from 1 day post hatch, so DPH to 40 DPH. Looking at, again, morphological, physiological, and behavioral endpoints with some visual assessment, which we'll talk about in a bit. But more than just documenting these physiological parameters, wanted to try and compare these 2 hatchery sites, finding again that one biomarker that's early on, that's measured early in development, that's going to be relatively predictive of that growth and success, both up to release and post-release. And again, that took place over about 3 stages each day. So the first stage was the respirometry. So again, putting these guys into these little glass well plates containing oxygen sensors to track their oxygen consumption and get that metabolic rate data over the course of a couple hours. While that was going on, I was putting more larvae under some microscopes there to look at those morphological and physiological parameters. So looking at body length and depth, fin ray development, opercular beat rate, and then some cardiological parameters that I'll discuss in a bit later. And finally, was getting some behavioral data by putting these guys into the swimming arenas, getting some top-down videos, and using the software called EtherVision to track their movement and get that parameter of average swim velocity. So, format of the figure a bit weird, but first thing we're going to talk about is that opercular beat rate. Because we saw, as we see, this is a pretty huge driver of what we saw later on in my data set. So, again, just measuring the rate of that movement is going to be a good proxy for the development of gill respiration in these fish. So when they're first born, these larvae are going to be mostly using that cutaneous respiration to uptake their oxygen. But that development of gill respiration is quite important because as they grow larger, the gills become far more efficient than that skin for the O2 uptake. So greater opercular movement, proxy for that development of gill respiration, is going to indicate that greater O2 consumption. Can allow those fish to have more energy available, more stuff that they can allocate towards those critical growth processes like digestion, etc. What we see is that those fish at the Corpus site were both exhibiting that opercular movement earlier at 5 DPH and exhibiting a higher rate of opercular movement across the entire study period compared to those Lake Jackson fish. Again, indicative of that likely earlier development of gill respiration, likely going to indicate that higher oxygen consumption, which indeed we see borne out in that data for oxygen consumption. So that was measured in both an absolute sense, so measuring that O2 consumption per organism, and a mass specific sense, so measuring that per unit body mass. But critically, the main thing we see here is that in that Corpus batch, those larvae were consuming quite a bit more oxygen for a given size, a given length fish, compared to that Lake Jackson batch across the bulk of that study period. Again, indicative of the fact that during those sort of middle to later development stages of those larvae, those Corpus fish were consuming a lot more oxygen, again, allowing them to have that greater growth scope, more energy that they can allocate towards those critical processes like digestion. Also borne out somewhat in that data of just their somatic development. So not as much of a huge difference here, but we do see that across the bulk of that study period, those Corpus fish were maintaining both higher length and depth compared to Lake Jackson. Although we see at the end here, that Lake Jackson batch actually caught up in terms of size with this Corpus fish. Talk about that a little more later. Also see in terms of fin ray development that those Corpus fish were developing fin rays in both their caudal and dorsal fin earlier than the Lake Jackson fish. That's going to be critical because, again, these fish prior to that metamorphosis, that settlement, they're very active pelagic feeders that will use that whole water column. So having those more developed fin rays means they're going to be able to control their movement better and be able to feed more actively. Again, critical towards getting that energy they need for growth in that larval culture. So overall, what we're seeing is that those Corpus fish are appearing to be a bit more developed in that sort of middle to later larval stage, roughly 15 to 35 DPH. And again, looking at that visual assessment of them, that seems to bear out. We see comparing that sort of 20 to 35 DPH, those corpus larvae in this batch were looking a bit more developed, a bit more tall bodied, more visible fins. That gut coiling was also a bit more developed in those sort of middle life stages. But a couple things we did notice. One, the chromatophores were more undeveloped in those corpus larvae at those later life stages. Not entirely sure why. That wasn't something that we were looking into for this study. But more critically, we saw that of those fish that were sampled at 40 DPH, every single one that I sampled at Lake Jackson was fully metamorphosed, fully settled, whereas not all of them were at Corpus Christi. But that actually might not be a big issue, because if those corpus fish are taking longer to get to that metamorphosis point, it could well mean that they're going to be larger when they actually get there and are released out into the wild. And that is borne out in terms of our body mass data. So again, looking at the dry body mass of these fish. So a small difference here is going to be reflective of a larger difference in that wet body mass. We see that the growth curve for that corpus batch begins a little lower than the Lake Jackson one, but right around 25 to 30 DPH overtakes it, continuing that trend up to metamorphosis and likely past that. And coincidentally, that time, 25 to 30 DPH, is where we also saw that peak in the auricular movement of the corpus batch. Again, when that gill respiration was likely fully developed at that point, where they were able to consume much more oxygen, again, allowing them to have that greater scope for growth, giving them that greater growth curve later on. So again, why does it matter that these guys are a bit larger? We do have some data on the survival rate of these southern flounder larvae in that sort of just pre-metamorphosis and slightly after metamorphosis stage. And the larger that larvae are, they seem to have a better survival rate in terms of both resilience to large temperature swings and salinity swings. So the larger release does appear to be something that might matter for their resilience. Then again, to corroborate that, we were looking at the cardiac performance of these fish, putting them under a microscope and taking some videos of the ventricle down here. So then using frames from that video to calculate the dimensions of the ventricle, and then using a process that's been used for zebrafish to calculate the stroke volume of that ventricle. So again, stroke volume being just the volume of blood pumped per heartbeat. Measure that, heart rate, and then the product of those two, cardiac output, or Q, just the volume of blood pumped out per minute. With heart rate, the only really notable trend we saw was that there is a large drop off in that heart rate right before and up to that metamorphosis and settlement. Again, it makes some sense. These fish are transitioning from that more active swimming lifestyle in that pre-metamorphic state, that more sedentary ambush-based lifestyle that they're going to exhibit throughout the rest of their life. The more interesting trend we saw was with stroke volume and cardiac output, where as you can see, these graphs are looking very, very, very similar for both stroke volume and cardiac output. That's likely indicative of these fish having a more volume controlled cardiac performance, meaning that when they need to modulate the blood flow, they're more likely to do so by adjusting their by adjusting their stroke volume rather than by adjusting their heart rate, which has not before been documented in southern flounder, but has been documented in a few other species of related flatfish, so it makes some sense. Also with stroke volume, we saw a couple more interesting things. So first off, just like how we saw that those corpus fish had that higher oxygen consumption for a given length, also seeing that for the bulk of that study period, those corpus larvae are exhibiting much higher O2 consumption for a given stroke volume. So for a given volume heartbeat, those corpus larvae were getting more oxygen out of it compared to those Lake Jackson ones for that study period. Again, indicative of that increased growth scope, because they're getting more oxygen, they can allocate more of it towards those critical processes that are going to allow them to go through that highly taxing period of development, especially in metamorphosis. Also seeing that when we look at stroke volume versus total length, stroke volume appears to be very well correlated, good predictor of length across both of those sites. So strong correlations, slightly better for total length, but also solid for that oxygen consumption. Meaning that stroke volume is looking like our best biomarker. Again, the goal of this was to find that biomarker measurable early on in life. That's going to be predictive of those morphological and physiological endpoints for these larvae. So we're seeing that with stroke volume. And again, these figures are a little larger than we were before, unfortunately. All right. So lastly, talking about that data on swim behavior. So we see that if we restrict our data set to only those fish that were fully metamorphosed at 40 DPH, so again, if not all those fish that I sampled at Corpus were fully metamorphosed, then all of them were at Lake Jackson. We see that those metamorphosed fish have that huge drop in that swim velocity around 40 DPH. with peaks in it right around 30 to 35. Again, not too surprising, given that these guys are transitioning to that more sedentary lifestyle once they metamorphose, but interesting to see. Then, if we include that full data set, so including all those fish at corpus that were not metamorphosed at 40 pH, we see that they're maintaining that very high swim velocity for all those later time points. Again, possibly a result of that fin ray development, allowing them to have that more free movement, more active feeding up in that water column. Again, correlating those morphological endpoints with physiological data that we're seeing. So, where does this leave us? Big takeaway from this is that that switch from cutaneous to respiration appears to be a huge factor in driving that growth for these larvae in that sort of middle to later development period. Again, enabling that more efficient respiration, greater food consumption, and we see that greater long-term growth curve for those corporate crusty fish. Also, again, that they were consuming that more oxygen for both the given size and that given stroke volume. Also seeing that, yeah, there's a pretty big variability in the metabolic rate of these larvae during that development period. Generally, we would expect that mass-specific metabolic rate to get smaller the larger the fish get. We're not seeing that for these larvae, so something interesting to potentially look at there. Also see, again, that stroke volume appears to be the best bet so far for that biomarker we can measure early on in flounder development. It's going to be indicative of their later on growth, health, success. As far as next steps, so it seems like gill respiration development is pretty important for these guys in that hatchery setting. So looking at things that we can possibly use to trigger that. So environmental factors that might be able to be adjusted in that larval culture that's going to lead to earlier gill development. Also, again, we're seeing that high variability in the metabolic rate. Might be looking at some energy budget modeling to try and see where that metabolic energy is being allocated, as well as looking at some metabolism profiles to see which metabolites are associated with various stages of that metabolic profile. I want to give huge thanks to everyone that helped me out on this, especially my co-chairs back at Texas in Galveston, Dr. David Holla and Dr. Lynn Pearson. Everyone in the lab that helped me out, especially to Libby Thibault. I've gotten so much good training from her, and to all my undergrad researchers that did a lot of great work with this data. Could not have done this without them. And of course, huge thanks to WD. Without that gracious funding and use of those facilities at Corpus Christi and Lake Jackson, obviously none of this would have been possible. So yeah, that's all I got. Thank you all.
Got time for questions?
What would be the differences between the two hatcheries practices that would cause you to see such a difference, like specifically in the oxygen rate?
Yes, that's definitely the question, right? So as far as the protocols, they are near identical in terms of how they're raising them, in terms of like the temperature profile, you know, when they start feeding them certain things. The main difference as far as the sites is obviously, you know, the location, you know, they're getting water from different areas, but as far as things that relate to that, that's not something that I've investigated yet. Yes.
Is there a difference in, or a correlation between the malpigmentation or the differences in the chromatophores with the?
Not that I could tell, but again, this is complicated by the fact that, like, you know, I wasn't getting all this data on the exact same fish. It was from the same batch, but once these fish go through respirometry, they're kind of done. A lot of that, like some of them will actually die in the process. So they're kind of not really very useful for other data that I want to get from there. They're too fragile at that level stage to really be resilient enough to get all that data on the same fish for each sample. Yes.
I think I just heard the answer to this question, but did you look at multiple batches from each hatchery, or were you just looking at a single batch in each?
Just one batch from each site with one caveat. So I did unfortunately get COVID during my sampling at Corpus. So for one of my time points at 15 pH, I did have to look at a different batch from Corpus Christi, but for the most part, yes, it was one batch from each site.
The reason I ask is because it seems like in our hatcheries, particularly for flounder, each batch has its own characteristics as far as growth and mortality and different life history parameters. And I'm just wondering how much differences in batches might have impacted the results that you're seeing between hatcheries, or even within hatcheries, if there would be differences.
Yeah, so that was one of the first questions that sort of led me to doing this work. So I'm building off of, like I said, my lab member, Libby Thibault. I'm sure any of you that have worked over at Sea Center probably run into her at some point. She did a lot of great work with the bioenergetics and more of the hormonal record analysis. So this study was sort of coming off of some trends that she found in the metabolic rate data from that Lake Jackson batch. So there is some data on previous batches that I'm using to inform this. But yes, for this chapter, for this study, that percent.
Yes.
I just want to reinforce, I think that that sampling multiple batches is going to be incredibly important. We found at the Corpus hatchery that there's a strong indication that fertilization success of a batch is indicative of an egg quality, not or egg or anyhow batch quality. And so that any particular batch, they do measure fertilization rate. And so if you could take a sample that had a strong fertilization rate and another one that had weak fertilization rate, you would have a range of variability of what I think is egg quality and see whether that shows up in your data set.
Sounds like, yeah, that sounds like a nice idea. Thank you.
Thank you so much.